In option 2, there are objective and challenges which include the need to construct the breeding strategy.
During the 4 breeding workshops that have been done, the backcross method, mutation and plant selection were the best method to include in this breeding design.
This is the overview question for option 2. So from option 2, we come up with 2 objectives, which are to retain the favorable trait of plant variety Y and also to improve the resistance of plant variety Y toward disease A.
There are 3 challenges that we found in option 2, which is number 1, there are no varieties with resistance to disease A. Challenges number 2, to have a variety that is resistant to disease A but still retain all the favorable traits of variety Y. The third challenges are, since there are no resistant varieties, the strategy that can be implemented is to create a new variety Y that are resistant to disease A while having all the favorable traits.
In this breeding design, the aim is to improve the resistance of rice variety Y to disease A while maintaining the favorable trait. Okay, so now we are talking about workshop 1. So in class, we learned about mass selection. So now we are thinking about, is it suitable for this plant breeding strategy? So the issue is that there are no variety that is resistant to disease A. So we cannot select any of the seed from variety Y. So we come to conclusion that mass selection is not suitable because the main objective of this design is to improve variety Y, which is resistant to disease A while retaining all the favorable traits of the variety. Okay. So now we are talking about workshop 2. In workshop 2, we learned about backcross method. We come up with the idea that backcross method is applicable and suitable for option 2, which is to achieve the first objective. While we need extra effort before we come up with this method.
During the workshop number 3, we learned about the mutation method. Mutation method is relevant for option 2 because we need a new variety of Y that have resistance towards disease A. After mutation method, we can proceed with backcross method. In order to do the backcross method, we need a chemical mutation such as ethyl methensulfonate (EMS), which is the most suitable procedure. We can obtain a new resistance of variety Y through this method. While during the last workshop, which is workshop 4, we learned about the plant selection method. The plant selection method is highly relevant because it is a reliable method to test a variety that performs well in different environments. The plant selection method can be carried out by using multi-environment trials of desired cultivars.
So what are the breeding strategy idea that we come with? So the first breeding strategy that we come with is mutation. Second is to do MET, which is multi-environmental trial, and then progeny testing, backcross, and also plant selection, multi-environmental trial. In this slide, we will talk about mutation more. So first thing first, chemical mutation. Chemical mutation, such as EMS, are most suitable procedure. This procedure will be done by killing 50% of the variety Y using various dosage of chemicals. A suitable dosage of the chemical will be used to treat variety Y. The treated variety Y will then be planted and grown. To test the effectiveness, the treated variety Y will be exposed to disease A. If there is a variety that shows resistance to disease A among them, then the variety will be
selected. Here is a brief explanation how mutagen works on this mutation method. The chemical mutagen mechanism involves EMS mutagenesis, which result in accumulation, whereby the original base is not physically altered, but the mutation is only fixed due to an error in the replication of the affected base. Guanine becomes replaced with adenine, and cytosine gets replaced with thymine. This disrupts the genetic code and can knock out gene function. The ethylation and base repairing error induced by EMS also frequently trigger the DNA repair system. The mismatch repair pathway recognizes the anomalies and repairs them. However, the The repair process is often error-prone itself, leading to additional mutation around the site.
Mutants that provide positive feedback will be chosen or selected through plant selection and then undergo multi-environment trials before the official selection. Multi-environment trials before progeny testing are a must to do because plant breeder need to perform the trials to get the information they need to allow plant breeder to make decisions. Multi-environment trials are not the breeding method itself, it is part of all breeding methods, intending to obtain a variety that can perform well in different environments. In this breeding design, about 4,000 individuals are required to start the mutation method, and then the population will be selected, will get smaller value but not less than 300 from each generation depending on the labor, cost and space provided by the plant breeder. In this slide, here are illustration of selected mutant that have to undergo MET. There are 3 generations of bulk harvest and bulk plantation of mutants. Each generation, the fixed mutation mutants will be selected to undergo multi-locational trials.
So after undergo the MET method, the next method is progeny testing. Progeny testing is a method used in plant breeding to determine the genotype of the plant by analyzing its offspring or progeny. It can be used to confirm whether a plant has a dominant allele that confers disease resistance. In our breeding design, we focus more on the GCA of the variety Y. The steps are by crossing the plant new release variety Y, RR, with the resistant with an original variety Y, YY that lack disease resistance. This is called a test cross. The first step is to grow the F1 progeny from this cross and inoculate them with the disease to test the resistance. If the F1 progeny are all resistant to the disease, it indicates that it is heterozygous with the dominant resistant allele RY. To confirm the F1 progeny is then be tested using general combining ability by crossing them with other susceptible recessive plant. If the F2 progeny segregate in a 3 resistant 1 susceptible ratio, it confirms that RR has the dominant resistant allele R that resistance is conferred by a single dominant gene. Based on the progeny testing, the newly released rice variety Y result in highest GCA values. We can assume that the genotype of the new variety Y with resistance to disease A has a dominant allele, which is RR.
In this breeding design, the aim is to improve the resistance of rice variety Y to disease A while maintaining the favorable trend. In this breeding design, the backcross method is applied to retain all the favorable trait of variety Y while being crossed with the new rice variety Y that has resistance to disease A. Assume that the genotype of the variety Y with resistance to disease A is RR, and the original of variety Y with no resistance to disease A is YY. The backcross breeding method illustrates the dominant allele for disease-resistant R from the resistant cultivar that is transferred to a susceptible original variety Y.
In this slide, here are the close-up of backcross diagram. In this diagram, there are 6 backcrosses, which is a new variety Y with resistance to disease A will be backcrossed with untreated variety Y. The ratio obtained from the backcross is 1 to 1, which is 1 RR, 2 RY, and 1 YY, with 98% of genes from Y, which is homozygous. This means that newly formed plants' genetic makeup become more stable and consistent.
We are now come to the last part of the breeding strategy, which is plant selection. Plant selection is an important approach used in plant breeding program to gradually improve crop attribute over successive generation. So the new variety will undergo multi-environmental trial MET in plant selection. The Finlay-Wilkinson regression model can be applied briefly to analyze the performance of the plant in multiple conditions. MET provide insight into adaptability, stability, and the link between performance and environmental factors.
In this slide, there are Finlay-Wilkinson regression model formula, which is P equal to alpha plus beta x. The data derived from the Finlay-Wilkinson regression model is then tabulated and can be converted into a graphical representation.
In this formula, alpha is equal to G indicate the performance. Genotypic performance across different environment can be visualized using a graph that illustrates the genotype by environment interaction. While beta is the slope, it shows adaptability or sensitivity to the environment. The slope represents sensitivity with smaller absolute values, reflecting stable response to changing environment. While E is the environmental index, which is an indicator of the quality of the environment.
Okay, to summarize our breeding strategy, so here it is. So the first one is mutation method, variety Y, undergo a mutation that may create a variety that is resistant to disease A. Second step is MET, plant selection, which we select the plant that is resistant to disease A. Next is progeny testing. And then we proceed with backcross method. New variety Y resistant to disease A will be backcrossed with variety Y that has all the favorable traits. The last step is plant selection, MET. The desired variety will undergo multi-environmental trial to choose the best seed which is stable and low susceptibility.
In our breeding design, we need to make the plant selection twice because at the first plant selection, we need to choose the best variety that is resistant to disease A before we proceed to backcross method.
The progeny testing must be done after the plant selection because we want to confirm the genotype of the new variety Y. The last step of plant selection, we have to do it again because we have to confirm whether the desired variety we want is stable and can adapt to different environment.
So in conclusion, genetic breeding program undertaken has successfully developed a new rice variety Y that retains favorable quality trait of variety Y while also exhibiting resistance to disease A. The 4 workshops during the class have provided insight to overcome the challenges and to achieve the objective by learning the suitable method to apply on rice variety Y. The objective of the plant breeding are met, which include maintaining all the favorable traits of variety Y and also improving the disease resistance to disease A. From this breeding design, we hope that we can obtain the best variety that maintain both objective and trait.
That is all from